Good morning, everyone. So my name is Barnaby Barnforth. I'm the CEO of a company called Biofidelity, which is working essentially to develop a transformational simplification of high-precision genetic testing. We were founded earlier this year actually as a spin-out from an existing startup company to exploit a novel piece of molecular biology that we had developed within this company and apply it to disrupting the genetic testing market. We, at the time, raised a £750,000 seed financing and were awarded a £500,000 grant. We also, as part of that, took in a small amount of investment from a US-based multinational diagnostics company who we're now working with to run a validation study internally. We're now seeking to raise a Series A financing of around £5.5 million, which will be used to develop sets of well-defined panels of genetic markers, which we can test for to guide therapy selection and for patient monitoring. And I'll describe a little bit about how that impacts patient management workflows. And then also to then get these tests to market through partnership with testing labs and with CROs, and to partner with pharma and kit providers to develop these into regulatory approved companion diagnostic assays. So what's the problem we're actually trying to address? There are currently more than 24 now FDA-approved therapies which are guided by molecular testing in oncology. At this time, fewer than 50% of patients who should receive molecular testing in oncology actually receive it. And fewer than 5% of patients are tested using high-precision, non-invasive molecular tests. The estimated cost to the pharma industry of that poor adoption of molecular testing is more than $8 billion. And the precision that is available by these really complex high-precision molecular diagnostics tests is ultimately required for routine testing of patients and for monitoring of patients and for accurate targeting of these therapies. The fundamental problem here is that the complexity of the workflows, the costs of these tests are just too high, and they are not suitable for widespread adoption. So this is what we have been working to address. So, one obvious, clear example is in non-small cell lung cancer. So there are 2 million new cases of lung cancer diagnosed every year. Only around 50,000 high-precision tests are actually being performed at this time. The precision of these tests is really required for accurate therapy targeting, for early detection of the onset of resistance to therapy while these patients are under treatment, and for monitoring of disease progression during treatment with these therapies. So there are big problems with the way this is currently being done. PCR tests are fantastic if you have one specific sequence that you're looking for and you know that there will not be a false positive due to background DNA. They are not suitable for this kind of testing. If I take a sample of blood from a patient, the vast majority of that DNA in that sample will be from healthy cells. It will differ by maybe only one base in its DNA sequence from the tumor DNA. A PCR test is not specific enough to pick out that single letter change. So the solution that almost everyone is working towards here is to use next-generation sequencing. Next-generation sequencing is fantastic as a discovery tool. It will give you a wealth of information. Most of the information that NGS tests will provide you are actually not needed for targeting these therapies. There are small panels of well-defined markers that need to be tested for, and clinicians do not care about the 100-page report that they get from these tests. They care about the markers that are actually used to guide these treatment decisions. So this is fundamentally the reason for the poor adoption of molecular testing. It's slow turnaround time, it's high complexity, and both of those translate into high costs. So this is where Biofidelity comes in. So Biofidelity was founded to exploit this unique piece of molecular biology that we have developed and to essentially enable routine genetic testing at this ultra-high level of precision, and to take these kinds of tests to the remaining 95% of patients who are not getting these high-precision tests in this very large market. So we offer a fundamentally different approach. We are not using sequencing. We have something that is a dramatic simplification of the workflows. So sequencing assays are slow, they are complex, they require a large investment in infrastructure. You need typically a dedicated lab which is just running this one sequencing assay. What we have is something that dramatically reduces the complexity of that. So a sequencing workflow will typically involve between 100 and 200 steps that need to be implemented or automated to take from a sample to a result. We have something that requires 4 steps. This is the point where most people will say, but the cost of sequencing is falling. It's falling dramatically, and won't that then solve everything? And unfortunately, the answer is no. The cost of the current sequencing tests is largely driven by the complexity, not by the raw sequencing cost. So the actual raw cost of generating sequence data only makes up about 25% of the cost of current sequencing tests. The rest is down to staffing, down to complexity of implementing these workflows. So we remove the need for sequencing. We provide ultra-high precision. In fact, we can detect single molecules of tumor DNA. And we provide that in an assay that can be straightforwardly implemented in essentially any clinical diagnostics lab around the world. I could put 4 tubes of reagents in a box, and I could ship it to any diagnostics lab, and they could run this assay now. So this is how we compare to sequencing. So with sequencing workflows, there are typically more than 100 steps. We have 4. The turnaround time for a sequencing assay is normally 1 to 2 weeks. There are some assays where you will get an answer in 3 days. Ours is less than 3 hours sample to result. The sensitivity is typically defined as this variant allele fraction number. So this is the fraction of DNA in a sample which needs to be derived from tumor cells for you to be able to detect it. And most sequencing assays will get around 1%. There are some that will get down to 0.25%. So Guardant Health is a very good example, gets down to about 0.25%. We can get down to less than 0.1%. And as I mentioned, we can actually detect a single molecule. The ease of adoption is key for us. We can put this into an existing network of labs worldwide. All they need to run this is a pipette and a PCR machine. It is not PCR, I should stress. This is a unique technology to us. It's very scalable. You can get hundreds of samples through on a low-cost instrument, and the COGS, the cost of goods for these tests, is dramatically lower than a sequencing assay. And we have a fantastic IP landscape, it turns out. So we have very good freedom to operate in this field. So I will very briefly touch on how it works. I'm not going to dive in deep into molecular biology. But essentially, we have discovered a particular enzymatic reaction, which is extremely specific to the digestion of double-stranded DNA, and it will only digest double-stranded DNA. And this is what we leverage to give us the level of specificity of our assays. So we design a piece of DNA which will perfectly hybridize to the sequence that we are trying to detect in a sample. We add that to the sample. It will hybridize both to the sequence we're trying to detect and to any similar sequences of DNA in the sample. We then use this enzymatic activity to digest only the double-stranded. So on the left here, where we have the mutant DNA in the sample, we are digesting the double-stranded part. On the right-hand side, there is a mismatch because we've hybridized to a healthy DNA molecule. We do not digest it. In that digestion process, we generate lots of these little stubs of DNA from the end. And that stub of DNA is what we can then amplify and detect. So we're leveraging this specificity of this enzymatic reaction to give us the sensitivity we need, and we are then doing an amplification at the end to give us a really high signal level. So what this gives us is a workflow that essentially goes from DNA extraction. We do an initial amplification of all of the DNA in a sample, and then we apply our technology. And during the final step of this, we are incubating the sample on essentially a PCR machine, and we are doing a fluorescent readout. And that allows us to detect and quantify the amount of target DNA in a sample. So the full workflow, including sample preparation, is less than 3 hours. This data analysis is extremely straightforward. Again, it's kind of like a PCR test. And we can multiplex this to large panels of mutations. So we actually took this and we went from one mutation, we implemented, I think I have this on the next slide, we implemented the full Roche-Cobas, EGFR and KRAS companion diagnostics panels. We actually did that in the space of 3 weeks, and we showed a 50-fold increase in sensitivity over the Roche assays. We've been working with the diagnostics company who invested in our seed round to do our validation studies. We are now showing single molecule sensitivity against panels of non-small cell lung cancer mutations. And that really now takes us to the point where we can move forward and develop this into real clinical diagnostic assays. So the IP position is very strong. As I mentioned, this was spun out from an existing startup company. We have transferred all of the IP related to this technology into the new company. It's owned outright. There are no restrictions or royalties. The chemistry behind this is unique. We have novelty confirmed by our examiner reports. We are proceeding with accelerated prosecution of our IP. We expect first grant of patents in the fourth quarter of next year. And we have an aggressive IP strategy to make sure that we are covering both the methods behind this, the implementation, the reagents, and then specific panels and applications for the technology. So our initial market is non-small cell lung cancer. It's a very large market, it's very well defined, it's poorly addressed. The low precision of current tests is a real issue. And the incoming guidelines for patient management are driving an enormous expansion of this testing market. So what we have is the precision and the simplicity that's needed to enable really routine testing of patients. And that's the way that patient management pathways are now heading. So this is kind of how these patient management pathways are evolving. So currently, if you present with non-small cell lung cancer, you'll be tested once. You'll be tested to try and decide which therapy you should be given as your first-line therapy. Where this is now moving is towards repeated testing of patients. So you'll be tested when you first present. You'll be tested repeatedly while you're being treated to see how effective that therapy is being, and then you will be routinely monitored after that period to try and detect when you develop a resistance to the therapy that you're being treated with. And some of these are already now coming in as recommended guidelines. So testing around every 6 to 12 weeks is now becoming recommended and is extremely poorly adopted. So our commercialization strategy is to essentially take this, develop these well-defined panels. We do not need to build our own diagnostics lab. We can partner with existing labs. We can build this into a workflow that is easily transferred into these existing labs. We can then get this broadly adopted really rapidly. We would define these panels from the beginning with input from physicians, with the reporting designed with input from physicians to make sure what we're presenting to the clinicians is what they want to see. And we'll take these two lab-developed tests initially through these partnerships with existing CLIA labs and with CROs for use in the biopharma industry. Companion diagnostics tests, I do not propose to go through the FDA regulatory proposal ourselves. We're a small company, and that's best done in partnership. And there is the obvious opportunity to build this into kits, which can be sold both for pharma research use and into research hospitals. So the beachhead application for us is non-small cell lung cancer, as I mentioned. As I mentioned, there's no need for building our own lab. We have inherently low operating costs. There are high margins behind this. We can build this as a revenue model, as a royalty model. We can get this really broadly adopted. One of the key selling points for us is our methodology is unique, which gives us the opportunity to go after a new reimbursement code in the US, which really would then give us a firewall against competitors and allow us to really drive adoption in these labs. So we can begin with a reimbursement code that's based on PCR, but the expectation is that this is differentiated enough that then we can get a unique reimbursement code for our technology. So we have a pipeline of assays that are under development. We anticipate the first of those being completed with external validation in the first quarter of 2021. The financing we're raising at the moment will take us all the way through to the end of 2021, which will allow us to get to the point where we have partnerships in place. We have some of these assays validated, both internally and externally. And we can then have these actually presenting results to clinicians. I will briefly mention the team behind this. Cameron is here, actually over in the corner there. Cameron and myself are co-founders of the company. We invented the technology behind this. We've, for the last 10 years, been building technology companies in the molecular industry. We've been joined this year by Heine Dreismann, who's former president and CEO of Roche Molecular Systems. He's extremely hands-on and has been very helpful in shaping strategy and in helping us drive this towards the correct applications in the clinical market. So if there's a message I want to leave you with, it's that what we have developed is something that transformationally simplifies molecular testing. We have something that eliminates the need for DNA sequencing within these high-precision testing for cancer diagnostics. We enable the earliest possible detection of these low-frequency biomarkers. We can detect a single molecule. You can't do better than that. We reduce the number of steps required from more than 100 down to 4. We can give a result in less than 3 hours versus days or weeks for sequencing. And we can scale this really straightforwardly on existing instrumentation. And fundamentally, it multiplexes incredibly well. We can build panels of multiplexed biomarkers, and we can apply these very rapidly to different applications. So what this enables is routine patient monitoring in a way that can be really broadly adopted, and this is a key requirement for incoming patient management pathways.